Okay, my final question in this section is explain what EPSP and IPSP are. Okay, so what are these things? Now, the EPSP is what's known as an excitatory postsynaptic potential, okay? And this moves the cell towards threshold by allowing positive ions to enter the cell as a result of the opening of ligand-gated ion channels, okay? So excitatory, just like it sounds, excitatory, is bringing the cell closer to threshold, okay? It's allowing positive ions to enter the cell, which means depolarize the cell, and which results from opening of ligand-gated ion channels. And the larger this excitatory postsynaptic potential, the more likely the action potential is to fire. Okay, and that's the goal. We want action potentials to fire in most cases. I mean, in some cases, of course, we don't. But I mean, a lot of times in an introductory course, you're thinking about trying to figure out how to get this thing to fire or not fire. But just like the wording indicates, excitatory, it's going to want to try to bring it closer to threshold and get an action potential to fire. And IPSP, you might have guessed by now, is what's known as an inhibitory postsynaptic potential. Okay? And these move the cell further away from threshold. Okay, so they actually bring the cell further from threshold, less likely to fire an action potential. Okay, and that's due to the movement of negative ions into the cell or positive ions out of the cell. Okay, so it could be one or the other. But essentially, the main point is it's bringing the cell further from threshold. Okay?